All right, thank you very much, everyone. Um, so today I would like to begin by acknowledging the traditional custodian of the land on which I'm joining today. I am based in the country of Wajak Noongar people, and so as the three speakers we got today. And I would like to pay my full respect to the elders, past and present. So today I would like to just provide a few updates from a conference committee, and then after that we move on to the talks that I put them in the program, and you probably saw those programs, and this is the reason why you're here for. So from the conference committee members, we are making a lot of progress to set up the programs and then to make this event very exciting. And one thing I would like to mention here is that the call for presentations are open now, and it is closing on the 7th of August. So please keep this important date in your mind and then make sure you will submit your abstracts on time. And then we are also seeking for sponsorship, and we found quite a few sponsorships has joined already, but then there are still opportunities. So please consider if that is something that you're interested in. There will be several travel bursaries, especially for ECRs, carers, and Indigenous participants. And all those informations are in the conference website, so please have a look. And I would also like to mention that we receive a lot of interest to host workshops, and all of them sound really interesting and very valuable. So whilst the programs are out, then you'll be able to have a look. We have a packed program, so please keep eye on that program release. And as well as the workshops, there are several social events, including ECR networking events on the first day, and then conference dinner. So
Okay,
yeah, we
Michelle?
are working really hard. I think someone is not muted. You're right? Okay. And yeah, so please keep eye on our programs, and we look forward to seeing you in February. So that was all from the conference part, and then let's move on to this talk. Sorry, I can't. Okay. So today's speakers, we got three speakers from Trend Lab in Curtin University. Dr. Alyssa Budd and Angus and Josh. I've worked with all of them back in the Curtin University or my current workplace at CSRO, and I'm always very fascinated with all the activities that Trend Lab have been doing. So today we'll be able to hear the research conducted over there. So we're gonna go in this order that you can see now. We start with Alyssa. She's going to talk about environmental DNA for natural capital accounting. So Alyssa, would you like to take over the stage? All for you.
Thanks, Mima.
Where is I need to stop sharing? Sorry. Yeah.
All right. Can you see let me put in presenter mode. Can you see my screen?
Yes, we can.
That's a good start. Yeah, thanks for the opportunity to speak today, Mima. So one of the projects that I currently work on is looking at how we can integrate environmental DNA into natural capital accounting. So one of the motivations, or the primary motivation behind natural capital accounting, is that economics has a blind spot. So in this image, you can see some logging of a mountain ash forest on public land in Victoria's Central Highlands. And if we look at this from a traditional GDP perspective, it looks pretty good. We get an increase in jobs and revenue from timber production. But what we haven't typically been assessing in many cases is the value of these forests when they're alive and functioning. So this might include things like ecosystem services, such as water purification, flood prevention, climate regulation, habitat provision, and of course, biodiversity. And what we often find is that the value that we might generate from harvesting our natural assets is far less than the value we would generate if we had kept them in their functioning or ecosystem service providing state. But you don't have to take my word for it. So McCormick and Showers did a study in 2019, or published in 2019, but over the 2017-2018 accounting period. And they were able to show that these forests are responsible for $806 million in water purification and provision to Greater Melbourne, 1.2 billion in erosion prevention, 356 million in carbon sequestration, compared to just 21 million in native timber production. So often people refer to natural capital accounting or ecosystem accounting as putting nature on the balance sheet. If you couldn't see these billions of dollars in ecosystem services, then 21 million looks pretty good. But once we compare these values, it starts to look a little bit less appealing to be chopping down these forests. One thing that you might notice is missing from this account is biodiversity. And we see this pretty consistently, that biodiversity is underrepresented in these accounts. So this image here is a lead-beater's possum. It's an endangered species and it lives almost exclusively in these forests. So activists were jumping up and down about it, but it didn't end up in the account. And a big part of the reason for that is that they're difficult to find. So you can imagine going out in this big forest, finding these tiny possums is difficult, and that's true for a lot of biodiversity. It would cost a lot of money to survey for one species, and if we want to include all biodiversity in many species, it becomes a bit of an issue. So I'm preaching to the choir here, but of course the opportunity here is in eDNA. So eDNA biodiversity assessments are incredibly efficient, and to illustrate this, I'm going to use some examples from the Trend Lab. So the first is Angus's Wetland Ecosystem Health Survey. So he's going to talk a little bit after me in more detail about this. But basically during his postdoc, he's been able to assess what looks like almost all wetlands in the southwest corner of Australia using eDNA. And Marina from the Trend Lab is planning to do a similar thing in the northwest corner. So it's kind of insane that we think these things are even conceivable in terms of project design, but the fact that researchers are carrying them out successfully is really quite impressive, and it just goes to show not only the great work by people from the Trend Lab, but also how efficient these eDNA biodiversity assessments really are. And so because of this efficiency, we think that eDNA assessments are going to be really quite good for natural capital accounting. So to illustrate what that might look like, I'm going to work through a bit of a thought experiment with you. And to do it, we're going to use the UN's System of Environmental Economic Accounting, Ecosystem Accounting, or more affectionately known as the SEA. So this whole bit of land is a bit of land that we steward as either a government or a company, and it's called our Ecosystem Accounting Area. We would split our Ecosystem Accounting Area into ecosystem assets based on the ecosystem type. So here we've got 3 ecosystem types. It looks like we've got a wetland, a desert, and a marine environment. And we would create a separate account for each of those ecosystem assets. But for the purposes of this thought experiment, we're just going to count the whole thing as one ecosystem type and asset. So to value our ecosystem asset, we could use something called the replacement cost method, which is the actual value of ecosystem restoration had we had mined or cleared this land for agriculture. And our imaginary value is going to be $1 million for that. And if we have something like Angus's Aquatic Ecosystem Health Index, we could measure the condition of our ecosystem asset. If it looks pretty good, we might be able to add a premium of about 30% to our initial value of $1 million. If we didn't have any sort of condition assessment tool, we could use something called SeDNAs. So that's taking sediment cores and analyzing the eDNA from those, which would allow us to identify pre-1750 conditions and compare those to the conditions that we see today. And that work has been done by Matt Campbell in the Trend Lab. We could also look at the presence of endangered species. So, for example, Ben Hayward from Trend has been looking at the Pilbara olive python with eDNA, and if we found that we had a bunch of endangered species, we might be able to add another premium, let's say 10%. Cheryl Lee Lucas from the lab does a lot of work on invasive species detection from eDNA, and in this case, we would be adding a discount. And then Nicole White does a lot of work on groundwater fauna. So these are ecosystem service providing species. And if we were able to get biomass estimates for these, so for example, from calibrated species-specific eDNA assays, we could then evaluate how much those ecosystem services will birth. So we end up with a total value of 1.25 million for our baseline, which is 2024. And then if we were to do this assessment again in 2025, but we've been good land stewards, then we might have an increase in condition, an increase in the presence of endangered species, a decrease in invasive species, and our ecosystem service providing species might be booming. And in that case, we can see that the value of our land has gone up quite a bit. And so it does provide landholders or land stewards quite a bit of incentive to take care of the biodiversity on their ecosystem asset. So thankfully, recently, we've just published some thoughts on accounting for biodiversity with environmental DNA in the journal Environmental DNA. So yeah, highly recommend checking it out. Just a bit of shameless self-promotion here. It's only about 1,500 words, but if you can't be bothered to read it, there's a pretty nice infographic that comes with it as well. So one of the challenges we identified when we were writing up this commentary piece is comparability. So it's difficult to compare eDNA studies across space and time. So to illustrate this, I'm going to use an example eDNA survey from Port Hedland, which Shirley is currently writing up at the moment. And it was quite comprehensive. So we have 5 habitats. We have intertidal reef, rockwall pylons, and then mangroves either within the harbour or outside the harbour. We had 3 to 6 sites per habitat, 9 water samples per site. They were sampling 1 litre of water through 1.2 micron filters. And I'm just going to look at the 16S fish assay, or they also did 18S. So I'm just going to use the data from this to show that eDNA is quite sensitive. So here we just have a OTU accumulation curve. So on the x-axis, we have number of sampling units or replicates. And on the y, we have OTU richness. And ideally what we'd see is that these curves eventually reach an asymptote with enough sampling. And we can say something like, okay, species richness is about 800. But what we actually see is that they just keep going up. So all we can say is that species richness is at least 600 and probably a little bit more for the rockwall habitat here. And we see this with most eDNA studies, or at least the ones that I've seen, is that more samples, we get more species. And that's even at very high replication. So for example, in this study, harbour pylons had over 100 replicates, and even then we're still climbing in this species accumulation curve. And what that means for us is that the richness that we're observing is actually a combination of true richness as well as largely sample coverage, but if we really want to get into the weeds, also detectability, evenness, etc. And importantly, it means that richness is not comparable across space and time unless sampling completely is similar or statistically standardized. So Anne Shao has a framework called INEX that tries to tackle this problem by using interpolation and extrapolation. I did try it on this data set, but unfortunately the variance around the estimates that we were extrapolating was too high to be comparable between habitats, but for other studies, it might prove quite effective. So what I've been working on at the moment is trying to identify useful metrics for natural capital accounting, where we do require these comparisons across space and time. And so I wanted to compare the robustness of different sort of traditional diversity measures to different experimental design and bioinformatic choices that I was able to change in silico. So the first is just replication. So I was comparing using 6, 12, and 24 replicates from that Port Hedland study. Minimum reads per replicate. So if the replicate had 1,000, either minimum 1,000, 5,000, or 10,000 reads. minimum OTU abundance per replicate, so either no minimum, 0.01%, which is commonly used in the literature, and 0.1%. And then also minimum OTU prevalence or frequency of occurrence. So if the OTU had to be present in a minimum of either 1, 2, or 3 samples to be counted in the first place. So we have 3 levels for each of these 4 parameters. So we end up with 81 combinations that we're trying to test for different biodiversity measures. And so this result is preliminary, but I wanted to show it to you anyway. So we're comparing 3 alpha diversity metrics in purple to 8 beta diversity metrics in green. And each point represents one of those 81 parameter combinations per metric. And then in the middle, we have the median values so that we can put our alpha and beta diversity metrics on the same scale here. And so what you can see pretty immediately is that the variability in alpha diversity, like richness or evenness, is huge when we vary all of these parameters, but far less so with beta diversity. So what this is indicating to me at this stage is that beta diversity metrics are probably likely to be much more useful for ecosystem assessments and natural capital accounting than alpha, or at least raw alpha diversity. Now, why is this important? I think that if you look at the ecological literature, people have probably been talking about this for a long time, but despite this, we keep trying to use it. So currently 20 of 23 examples of ecosystem condition variables listed in the UNC are richness-based, and one of just 2 eDNA methods in accounting for nature's methods catalogue, which is specifically recommended by the TNFD, or Task Force for Nature-Related Financial Disclosures, is also richness-based. So these documents are the current leading global frameworks for natural capital accounting and what that might look like. And TNFD specifically is likely to become mandatory in Australia in 2028, although it won't be called TNFD, it would probably be called Schedule 3. So I think we really need to be thinking about what measures we're going to be assessing the environment with, and whether or not they're robust and really can be used for accounting, particularly comparing the same ecosystem across different time periods. So just to wrap up, we really do think that eDNA is likely to be useful for natural capital accounting and ecosystem accounts because of its efficiency. But comparability is a key challenge. And I just wanted to point out that this is not necessarily limited to eDNA surveys, although they are quite sensitive, which makes things tricky. It's also why they're powerful. But biodiversity is just really tricky to measure. And then the final point is that I think more research is needed to identify appropriately robust biodiversity indicators that we can really be sure that the measures that we're generating are going to be useful for natural capital accounting. So I hope to continue this work, but I also hope to see more examples in the literature pop up of other people thinking in this space as well. And that's me. Thank you very much.
Thank you very much, Alyssa. It's really exciting. Congratulations on the paper. I'm definitely gonna take a look at it straight after this today. And yeah, it's really exciting to see that you established this on the huge efforts that have been happening in the trend, like multiple people in the Trend Lab, including Angus' work that he's going to present now. So I can see like how important these individual works are to build this kind of like a big index. And
yeah, for sure.
Yeah. Okay, so we can ask the room if anyone has questions to Alyssa. Yeah. Does anyone have a questions from this talk? Olive posted something, but yeah.
Yeah, I posted something. So you can either read it or I can ask.
Yeah,
Alyssa,
or you can ask.
Great talk, Alyssa, as usual. What if alpha diversity is so problematic? Why is it so widely adopted when there are alternatives? Like what's the disconnect?
It's a great question, Ollie. I think it's easy. It's a single value that can be very easily interpreted. So it's number of species, easy for management to read. So a lot of the time when we look at beta diversity, it's a matrix of things, and we're kind of looking at, something on an NMDS plot. So part of the metrics that I'm developing is collapsing beta diversity into a single value that is a bit more easily, maybe not interpretable, but at least usable by management. But I think primarily it's just easy to get, it's easy to interpret and people understand it. And I think there are some ways around it. So if we did occupancy modeling and tried to figure out kind of error bars around that richness, so we actually knew if richness of 10 and 20 was different between studies based on variability, then we could probably use it a little bit more effectively. But I think the way that it's currently used, at least cited in these frameworks, I think it's quite problematic. But yeah, I guess short answer is it's easy.
Right, thank you. Does anyone have any question? If not, then we move on to the next speaker, who is Angus. And so he's going to talk about something that Alyssa touched a little bit. So it's on the eDNA taxon independent index for biomonitoring of wetlands.
All right.
The stage is yours,
cool.
Angus.
Great. Good day, everyone. Thanks for having me. So I'm a molecular ecologist. Most of my work focuses on wetlands and different inland water bodies. But I guess I'd like to start my presentations by acknowledging that wetlands are really important ecosystems in lots of ways that aren't always front of mind. So they give us our water purification, they prevent damage from floods, they're critical in the recycling of nutrients, and they're actually a really effective carbon storage. They contain and support vast numbers of endemic and endangered species. They're especially important in dry environments like we have in the southwest corner of WA, where they support a rich variety. Sorry, I'm just trying to switch slides here. We have a rich variety of different types of lakes. So they come in, they're big, they're small and temporary, they're luscious, or they're salty, and they come in a range of different types and forms, and they're really heterogeneous environments with very different communities in each of them. But generally, what they all have in common is they support a variety of different animal types and plants. I'm going to focus on animals of all manners of different kinds. My favorite, just for clarity, is the ones on the far right, the brine shrimp, which I love very much. But despite their importance and their significance, they face numerous challenges. A great statistic that I like to cite is 70% of the wetlands that are on the Swan Coastal Plain, so the areas around Perth, have been destroyed or infilled since colonisation. This is actually a mine on Rottnest. This is a limestone mine. They're drying out as a result of climate change, and in the southwest of WA in particular, we have significant impacts from agriculture, which leads to very intense salinisation. So these environments are, significantly at risk, and we obviously need different ways of managing them. One of the interesting spaces where I think the intersection of eDNA and biomonitoring comes in is in relation to some of the things Alyssa's been talking about, is that when we're calculating indexes for how different environments are going, we're effectively just making observations regarding what taxa we find where. I always get a bit of stick for this graphic because I put the vertebrates on one side as good and the invertebrates as bad on the other side. But for diagrammatic purposes, effectively, when we define most indexes, we tend to just base our observations around where certain taxa occur. So in this example, a good lake might represent fish and a frog, and a bad lake might represent only worms and more hardy taxa like snails. But as we all know, what each of these taxa are leaving behind as a result of their presence will be their DNA. But why that's particularly useful, I think, is because we're able to build a more holistic picture of all of the organisms that are in each system, rather than relying on typical observational data, which might lead us to counts of single digits to tens of observations. DNA obviously allows us to have thousands or hundreds of observations per ecosystem. And this project is focusing specifically on leveraging all of the DNA that is available to us. So we're not restricting ourselves necessarily to the taxonomically identifiable sequences, but rather the whole piece of the puzzle, which, as we all know, when we're working with DNA, is often the majority of DNA sequences. So effectively, what this study and the postdoc that I've been doing is trying to do is answer four, predominantly the first question at the top, but it leads into the other four. I'm going to step through these questions as we go through. But effectively, what we're trying to design is a taxonomy-free eDNA index that we can use to track anthropogenic pressures for wetlands in southwestern Australia. One of the questions that's dawned on me as I've done this exploration is we're working with a very large data set. I've been very curious around the actual applicability of this index insofar as the financial constraints when you're using eDNA. So if you have to use heaps of heaps of assays rather, is it actually economically viable, or is there a more economically viable way to get the same answer? Then, how good is this actual index? One of my key frustrations is people not critically evaluating the indices or being upfront with the limitations of their indices that they put out to managers and not communicating their limitations more effectively. So I'm going to go through that. But effectively, what this study has involved is a lot of data collection. So initially we were focused on areas around Perth, deliberately limiting our sampling to a single bioregion to deal with issues of whether or not DNA sequences collected from one biogeographic area might be distinct from DNA sequences collected from a separate biogeographic area, and whether or not that would affect the model or not. It doesn't seem to, as a long story short. So then we expanded our sampling to the rest of southwestern Australia, and we ended up with this very large data set that covers wetlands of a variety of types. It focuses mostly on freshwater wetlands, although I deliberately included a bunch of saline lakes, which I'll talk to in a minute. We then blasted these water samples with 13 metabarcoding assays, ending up with 6 replicates times 171 wetlands times 13 assays it's a lot of data. We then pumped this large data set through an unsupervised learning model called the Chessman iterative process, which I'm not going to go into, but effectively all it does is, the only critical thing to understand is I've told the model nothing about the precondition of any of these sites. It's effectively a complex ordination method that blah, 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 blah. I'm not going to actually bother going through that. It's quite complicated. If you'd like to know, I'm quite happy to discuss it later. So we used all this information and we built this model. And then the secondary question is, okay, we know that these wetlands change substantially throughout the year. So how does the index change throughout the year? Because you can imagine that if your index varies based off individual sampling points, so if we go in March and we get one value, or we go in June and we get a completely different value, what does that kind of context actually mean? So I'm still in the throes of finishing all this lab work, so I actually don't have results for this at the moment, but this is just to point out that we have a very large spatial study, and then we have a very detailed temporal study where we've sampled 11 sites for a year. And these sites are both permanent and seasonal, and we're interested in the effects of do the kind of complex seasonal changes that we observe in wetlands in southwest WA have an impact on the index that we're producing. But the actual model, so just answering this first initial question of can we actually build a model that works? Before I put the results up on the screen here, I just want to explain the Y and X axes. The Y-axis is the aquatic index, so this is the actual model that we've made. But the X-axis is essentially this kind of independent assessment of wetland condition, and that's a series of loaded terms. But effectively what it is at each site, I've done an assessment of how polluted is the site based off its amount of nutrients, how many invasive species does the site have, what is the proportion of natural cover around each site. So you can imagine that's like the riparian zone. So that effectively the eyelashes that keeps the dirt out of the lake, assuming that if you have a lake with lots of natural remaining vegetation, it'll be better condition than one with none. and then also whether or not a lake has experienced any known hydrological changes. And that can be going from permanent to seasonal, or from seasonal to permanent. So a change in how much water and how long it's holding that water for. And then effectively, I gave each lake a score out of 25. And then the x-axis is effectively the value of how disturbed each site is. So if this model works, we'd assume a linear relationship. Oh, yeah, great. So effectively what we've found is the aquatic index that we've developed, based off the DNA sequence data, seems to have a quite strong relationship with sites that we'd expect to be poor. So sites that have high amounts of nutrients, lots of invasive species, and have had a fair proportion of their native vegetation removed, tend to score worse than sites that have the opposite. Obviously, that relationship's not perfect, but for ecology, I think it's a relatively good relationship. How you actually then interpret a score of 25 versus 75 or 50 is a slightly different conversation, and I'm still kind of working through the framing around what it means to be very poor, moderate, good, excellent, because obviously where you put those different parameters, it has an element of subjectivity to it. But broadly speaking, we're relatively pleased to find that there is a relationship between sites that we'd expect to be bad do score poorly and sites that we'd expect to be good do score well, and that this doesn't necessarily seem to be affected by biogeography. So a site in Perth is just as likely to score on this scale as a site in Albany and anywhere else you could find in the southwest. So just to reinforce that point, these are some of those characters that I was describing before. So this is the amount of natural vegetation relative to the index. So again, on the y-axis is our health score. So sites with lots of natural vegetation score better than sites that have low amounts of natural vegetation. Sites with large numbers of invasive species score worse than sites with no invasive species or less invasive species. Sites with lots of nutrients score poorly. So these are just the kind of extra little steps of understanding what our index may actually be responding to. But this is the question that I'm kind of interested in articulating today, which is, okay, great, you've got 13 different metabarcoding assays. To do that for an environmental practitioner costs a lot of money. So if you want anybody to actually use this index, how can we make this more cost effective, or how can we make this more efficient? And the good news is you absolutely do not need 13 different metabarcoding assays in this model. So effectively what I did is I looked at the relationship between the aquatic index score and those independent metrics of environmental health that I was describing before, and I looked at the strength of the relationship between different combinations of assays together. So this goes from 1 to 13. And what we tend to find is effectively there's no difference in the performance of the indexes that you make once you get past about 3 assays in a model. And that essentially means that there's a lot of redundant information as you add extra assays into the model, which makes a lot of sense, because if you're measuring community composition, I think so long as your assays are relatively universal, you're having similar patterns across multiple taxonomic levels, which is my current working theory, but I'm still kind of exploring that at the moment. The other point is to say that you can get relatively good models with single assays. They're a bit more variable, but they work relatively comparably well as combining multiple assays together. But some assays obviously do work better than others. So all you need to realize here is that the 3 green ones are the 3 assays that are ending up in the kind of, I guess, final model that we're working with at the moment. And you can see that their performance is roughly comparable, but they're roughly comparable with a lot of other individual assays. So there are lots of correct choices to be made here and very few wrong ones. The next question is, I guess, how repeatable is the index? So how many replicates do you need to actually get a reliable score? Because again, replication is another thing that contributes to cost. So how many assays you have and how many samples you need to take. What the interesting things is, I find quite hard to articulate specifically why this is the case, but for the reasons for the way that the unsupervised learning model works, and if we remember our central limit theorem from second year statistics, which I'll get into in 1 second, we actually find that you don't need that many replicates to get very close representations to a site's true score. And the reason for this is how the model effectively works is it assigns a value to each unique DNA sequence. And you can imagine that these values they follow a normal distribution. And so if you collect a large number of DNA sequences from an individual site, you're going to end up pretty close to the true value. So you can see here in this plot, what I've done is I've segregated the amount of indicator sequences that are found in a replicate, and then the differences from left to right are how far away that score is from the true site score. And what we find is once you have more than about 20 indicator sequences in a sample, you get pretty close to the actual true aquatic score when you base it off all of the available indicator sequences for that replicate. And that's probably why we're able to get away with relatively low numbers of replicates. But anyway, this is still a bit of a work in progress. And then finally, what are its limitations? The main limitation that is relevant to South and Southwestern Australia at the moment is that this model does not work on saline lakes, and there are lots of saline lakes in Southwestern Australia. The reason it doesn't work on saline lakes is effectively all the model is a really fancy ordination method, and it's relying on the assumption that your species are different from each other. Sorry, your communities are different from each other, mostly driven by some form of environmental disturbance. Where you have really strong environmental factors that change community composition, so for example, going from a freshwater lake to a saline lake is a huge difference in its overall community shift. Those large environmental shifts override any underlying effect of disturbance because they're just a bigger shift in community composition. So you end up with poor relationships between how poor you'd expect a habitat to be and how poor our model thinks it is. So this is an important limitation, I think, to be very transparent around where this model can be used and where it can't be used. And then just wrapping up, some more limitations. The other thing that I think is quite interesting about working in wetlands, and I think this applies to lots of aquatic environments, is, and particularly when we're working with dark eDNA, so we don't necessarily know where it's come from, it's very important to be upfront with the fact that a lot of the eDNA signal that we're measuring probably comes from terrestrial sources rather than exclusively from within the wetlands themselves. For the validation of the index, I'm not at this stage not necessarily considering that as an invalidating feature, because to my mind, wetlands act as kind of sentinels of the overall landscape, and a wetland health is intrinsically linked to its catchment. So if we have a different community of organisms flowing in from outside sources, I don't necessarily think it matters whether or not they came from the wetland themselves or they came from the input from around the surrounding terrestrial environment, because I still think they point to the same direction. One thing I do think is important to recognize in that space, though, is that if you consider disturbances that might uniquely affect a wetland independent of the terrestrial environment, so if we take potentially nutrients as an example, you can imagine a situation where the aquatic life of a wetland will be affected by increasing nutrients, but that won't necessarily be reflected in the terrestrial environment around it. It might be in some circumstances, but not always. So is there a disconnection between the lake effect and the landscape effect? It's tricky to answer that question now, because I think validating these types of indices takes repetitive sampling over years to understand whether or not and how these indice values change in response to disturbance events. So just generally wrapping up what I've just said, we've made a model that I think works reasonably well for tracking broad-scale anthropogenic pressures for wetlands. You definitely don't need to throw thousands of dollars at using this model. I think it actually is a relatively cost-effective way of getting a quick triage of how a wetland is going. And though we still have important questions that I need to find answers for in terms of where can it be used and where shouldn't it be used, which I think is really critical before we roll this out to any form of environmental management group, that won't necessarily ask those questions, but will rely on us to have given them something that is fit for purpose. And the other thing is, if anyone's got any cool ideas to do with lots and lots of eDNA data, please get in touch, because I lack the imagination. So thank you very much.
Thank you very much, Angus. Great talk. I don't think you lack any imagination. Your imagination was put all into this presentation and on the project, and then we were very impressed. I can see Ollie putting a couple of questions. Would you like to? Would you want me to read it out, or maybe you can?
So it's what is driving the variance in the index? So it's a really good question. The things that are driving the variance in the index is the extent to which two habitats are different from each other, not due to anthropogenic impacts. So you can imagine, for the same reason why salt lakes and freshwater lakes are widely variant in their communities, are the same ways that eventually, if you went far enough away in a biogeographic context, I believe that the model would stop working. Because as soon as essentially, you can envision what the index is doing is you have a soup of DNA sequences that compromise your community, and you're assuming that there is an opportunity for all of those or some of those DNA sequences to exist between different sites. But if those sites are different for reasons that are completely unrelated to disturbance, then you can't get that relationship. You won't pick up a marine fish in a freshwater lake, or you might from bird shit, but you won't in reality. The biogeographic factors one is really interesting. I've been thinking about this biogeographic thing for a while, because my initial concern, which is why we limited it to quite a small spatial area initially, was because of biogeographic impacts that I was worried about. One of the things I think it's interesting to think about is that if you're using, it depends on what gene you're working with. So you can imagine that if I'm working with CO1, or I'm working with some 16S, but if I'm working with 18S, if I move biogeographic regions, I'm going to end up with the same ASV between biogeographic regions, right? If, for example, I'm describing, I like to use the example of dragonflies. So we know that there are dragonflies in lots of different places across Southern Australia, right? And they might be different genera, maybe they're different families. There are representatives of those different families in all regions. If we move large spatial distances, it's likely that in the conserved ASVs that we're working with, they might be retained across both regions, and therefore retain the same. We would still pick the same ASV up across large spatial areas. Whether that applies for some of the other more quickly evolving genes or not is probably not true. I think there are lots of ways you can fiddle with the outputs of these indice values to scale them for different known effects. So you could weight different indice scores in certain situations where they were more or less important depending on where you were in the world. The other way of doing this, of course, is you just do a supervised learning approach where you say these lakes are good, these lakes are bad, these are the ASVs that are associated with these two different environments. I was reluctant to do that in the first instance because I didn't want to impose a somewhat subjective criteria on what was good and bad. But effectively, we've ended up doing that anyway, because the way that we're validating our index is with an independent assessment of wetland health, where we are ultimately scoring these lakes based off a certain criteria anyway. I think that's a broader discussion around whether or not you should be using unsupervised or supervised learning approaches in developing in using biomonitoring indexes for eDNA data anyway. I think that's quite an interesting separate discussion.
All right, thank you very much, Angus. I can see Ollie still typing, but I'm going to move on to the next speaker, Josh, because we got a bit over 10 minutes left. So Josh is going to speak about his project on exploring eDNA in a terrestrial ecosystem. So we're moving from water samples in a wetland to terrestrial environment now. Josh, please share your screen and then go ahead.
Cool. Can you see that now?
Yeah.
Yeah, cool. Thanks very much. So yeah, I'll take you all onto land now and chat about kind of what I've been doing over kind of last 4 or 5 years, just a few different projects around air DNA. But for all of you guys that are more water-based, I thought I'd just touch on why in a terrestrial system air DNA is, I think, quite important. And for a lot of years, people were basically using water. I'm kind of going to speak in the context of vertebrates and plants for most of the talk, but people have been sampling water to try and detect terrestrial vertebrates and then soil as well, which doesn't work too well. Then we started looking at in kind of swabbing surfaces and stuff like that. But about 4 or 5 years ago, some people started to look into the idea of collecting the DNA from the air. And I think that created a bit of a fundamental shift in terrestrial vertebrate DNA kind of space, because all of a sudden, like water in aquatic systems, you've got a substrate that's now surrounding every organism that you're interested. So it's not biased in any way. It's quite simple to collect, and you can collect it absolutely anywhere in the world. But what a lot of people were, what they started with when they were looking at detecting vertebrates, what they were looking into is using kind of computer fans and different filter systems to capture DNA onto filter paper. So our first delve into the world of air DNA, we looked at spider webs. We wanted to trying to think of a simple substrate in the environment that might be capturing air DNA, where you don't need any complex equipment or anything like that, and you can just go into the environment and sample. Really simple. We went to a woodland and a zoo area, and we just we got some swabs and we collected spider webs. I liken it to when Princess Fiona makes some fairy floss for Shrek out of spider web, just wrapping it around a stick. And it worked really, really well. We collected it. We got a heap of zoo species, lots of mammals, lots of birds, not too many reptiles, which is pretty common, and not too many amphibians, which would be expected. If you're on amphibians, you're better off sampling water. But I think the kind of main takeaway from the spiderweb work wasn't necessarily that we should all be going out collecting spiderwebs if we're looking for vertebrates, but the idea that air DNA can be passively collected onto a surface. And spiderwebs are more of a benchmark for what a good passive capture surface could be that we should kind of be looking into. So as an extension of that, we decided to have a look at using powered systems like other people have been working with, and then comparing that to kind of passive, and then a more trying to use the wind to our advantage. So we went to the same 2 sites, which is Crackamire Wildlife Sanctuary up in the hills of Perth and the Perth Zoo. We set up this system around the place where we had the passive, just a bit of filter paper literally hanging in the breeze by a peg. And then the powered system, which is a 3D printed little device that goes onto a computer fan, and it's run on a battery, runs for about 40 hours. And then a kind of weather vane kind of device trying to capture the wind and get more directional input onto our kind of filter paper, all the same filter paper, and everything was left out for 48 hours. And what we found was the active sampler was by far the most efficient way in a short period of time to capture air DNA. Spiderwebs were pretty comparable, especially at Crackamire. They were our kind of second best substrate at both sites. And I'd kind of call spiderwebs probably a long-term passive. The other passive samplers were only out for 48 hours, so leaving them out longer was obviously much more efficient. So spiderwebs are good, but passive worked, and they worked okay. Just more work needed, kind of probably longer exposure times and more development on what we're using to capture the DNA on. But interestingly, the wind one that I spent the most time developing didn't work at all. And I think the lesson there is that we actually, instead of enhancing the DNA capture, we actually reduce the amount of surface area that's exposed to the wind, and so significantly reduce the amount of DNA. So if you are looking at doing passive sampling, you're actually better off probably just going out and hanging a bit of filter paper with a peg out on a twig instead of trying to develop any fancy devices. The take-homes from all of that were really active sampling. You got the highest richness over that really short deployment time, so best for short window operations. and low airflow sites. So if you're going into caves or tree hollows or anything like that, you'd probably be looking at an active system. But the downside of it is it needs a lot, you need power to generate it, and they're a lot more expensive to build. So your higher cost and limited run times as well, I think they really cap our ability to go at scale and cap replication. Whereas for your passive sampling, when it's optimized, because it certainly needs to be optimized, it's really simple, scalable, and extremely inexpensive. If you're going into remote areas, you just got a bag full of filter paper and you can go peg them up everywhere. So great for large scale kind of things. But then you do run the risk of you're going to be leaving them out for much longer, so potentially DNA loss. And if you get something like a rain event, we know that washes away a lot of DNA, so you kind of have the risk of losing DNA. There is a major shared limitation, which I think is a pretty large downside to both of them. And it's that you have to deploy your sampler and then you have to go back to the same site to retrieve your sample as well. eDNA is supposed to be this kind of rapid, scalable tool. And I think if we want to go large and we want to go over real large scales, then having to go back to your site to collect your sample makes it a bit more complex and less usable for practitioners. And we also don't want to be going out and collecting spider webs everywhere, being our eDNA source, where we can just go on a single visit. So we then started to think about how we could sample at scale and rapidly sample. And this is where we kind of looked at the idea of, can eDNA actually be collected while we're moving through the landscape? So what if we had a filter paper that's attached to a car or a drone, and then you're putting them both together. So you, in some respects, actively sampling because you're using the wind to push through, while you're driving, to push through some filter paper. But it would be just a single sampling event as you're driving through the landscape.
Yeah, I'm just reconnected. I think it's just.
So what we want to do, though, is go at scale. If we're going to be doing air sampling with a car, can we go and do a huge spatial scale? So this is southwest Western Australia. And what we decided to do was to drive all the way from Perth down to Cocklebiddy, taking samples around about 26 kilometres. You can see the sampler on the right-hand side is basically just some aluminium with some magnets on it. And we used a histology case and some histology foam as our air filter, which tended to work pretty well. So you've got the opportunity to literally at 110 k's an hour be sampling your eDNA, just taking the filter out, and then you don't have to stop to change over the filter, and just rapidly sampling through the area. By far the most boring field work I've ever done in my life, sitting in a car with two middle-aged professors for 4 days. We're driving from Perth to Cocklebiddy, so 2 days there, and then we turned around and we drove 2 days back, sampled there and back. What's really important about this area, though, is it's got some really distinct biogeographical breaks in it, because I think one of the biggest unknowns with air DNA is how the DNA is travelling through the air. We know that it can travel for pretty substantial distances. There was some work in Great Britain where they had their mean detections of being about 18 kilometres away, and then maximum detections being about up to kind of 80 kilometres away. So small scale studies using air DNA potentially going to be quite limited. But if we go large scale, is this just, I suppose, a mix of just DNA soup through this area, or can we actually pull apart kind of ecological, meaningful differences in our samples? So we started in the Swan Coastal Plain, the blue area, drove through the Jarrah Forest, which is the pink, and then into the Avon Wheat Belt. So for anyone that doesn't know WA, this is our extensively cleared agricultural region, basically just wheat and sheep farming area. And then we get the green, which is our Coolgardie bioregion, but the Greater Western Woodlands, hugely biodiverse, incredible bush woodland. And then down over east, we start to hit kind of an ecotonal region between the Coolgardie region and the Nullarbor Plain, so getting into more grasslands again. We ran 3 assays against this, looking for vertebrates, invertebrates, and plants. And what was great for us is we actually see really distinct changes in our community as we're going from east to west. So what these plots are, they're just PCOA plots. Axis 1 is colour, so blue to red, and axis 2 is the size of your point. So anything that's closer in size and closer in colour is a similar community, but this is just plotted through space. We see, obviously, in your agricultural region in the yellow, you get these real kind of similar communities. Hit the greater Western Woodlands and you get a lot more diversity through there. And then down on the bottom area where it starts to become a bit more kind of grassland, it becomes a bit more homogeneous again. But I suppose the main question is, if you get a clear east-west pattern, is it just driven by distance, or is it actually driven by environmental gradients? So we did some general dissimilarity modelling on all this data to see what environmental drivers are maybe driving this community turnover. And excitingly, it wasn't just distance that was driving the community turnover. For plants, it was rainfall, precipitation that's driving the community turnover. Explained about 36% of our model. This is by far the strongest signal we got, and that is all very much in line with kind of previous literature as well, which is great. Vertebrates was driven by temperature and habitat type as well. The model wasn't as strong as plants, but I think because we had vertebrates all lumped together, if you separated your vertebrate groups and you had some more samples, I think you would have got a stronger signal. But definitely still, it's your climate and habitat types that are driving your vertebrate groups, which is great. And again, not distance, which is important. And then for our arthropods, this is our weakest model. And I think just because we detected about 600 insects in a region where there's about 13,000. So we don't have the data to really pull apart the insects, but we can tell that there's changes in community every about 50 kilometres for our insect groups. And it's likely because the environmental variables we were looking at probably are a bit too coarse for vertebrates, that are driven by microhabitats and things like that. But the other thing we looked at with this data is we looked at some high impact species. So these are pest species listed as pest species. Some are native and some are non-native. So you got rainbow lorikeet, we've got a stable fly, galah, and an emu. And these are the GBIF records of where these animals, distribution of these animals, and the orange overlay is our eDNA data. And what's great is they're kind of all popping out exactly where you would expect to find them. So you're not getting the DNA of these species travelling everywhere. For the emu, I've never seen an emu around Perth, but where they're being picked up there is all in the northern kind of greater Western Woodlands. Same for our rabbits and our fox as well. So rabbits, as you would kind of expect, picked up all through the data set, and foxes just really picked up in that region where you can see the distribution of the fox. So I think really exciting, and what it's all kind of showing is the atmospheric movement isn't creating this kind of big soup, but we're really getting a true ecological signal within our eDNA data. Going kind of back on what Alicia was saying with richness being potentially an issue, but the usability of beta diversity, I think that eDNA data from the air, and I think when we talk about airborne DNA, it's actually probably every substrate that we're collecting in a terrestrial system, because even if I'm swabbing a surface, I'm still picking up DNA that has moved through the air. There's definitely the potential for DNA to be travelled over a really large distance, but the underlying community that we're detecting, I think, is a true ecological signal, which is really important. So where do I think kind of the usability of AirDNA might be? I think small scale studies using AirDNA, I'm not 100% sure if they're going to be usable at all, just because of the movement of the DNA. But large scale monitoring tools where we can do a real big pass of an area at scale could be really valuable. For tracking biodiversity or detecting, pest species, invasive species at the front of an invasion, I think it can be could be a really valuable screening tool to help decide where, when, and how to kind of direct our conventional methods. So instead of, if we're trying to detect an invasive species in the whole of southwest Western Australia, where do you first go to target, and where do you put your people on the ground? If we can run these big eDNA surveys, just sampling the air, and try and get more of an idea of where to put our money and where to send our people onto the ground, I think it could be really valuable. So thank you all very much.
Thank you very much, Josh. It was great talk
You're
again. Yeah, very exciting to see where this airborne DNA is going. I remember few years ago, we were all doing this kind of experiments in the water environment. And now obviously we all realize that air can be very useful, and then you are proving that. So it's very exciting. Unfortunately, we don't have time for the questions, but if you have any, then you can probably email to Josh directly. And so I would like to close this webinar today. Thank you once again for the speakers to provide amazing information from your works. We all learn a lot today from you. I would like to just mention quickly about the next webinar. So we have this webinar about every 3 months, and the next one will be in October. And we got 2 speakers lining up. They are John Piernan and Georgia Thompson-Lang from both are from Cochrane. So we're moving from Trent Lab on the west coast of Australia all the way to New Zealand to Cochrane in the next webinar. And we will send you the registration link and things like that in the coming weeks. So please keep your eyes out on the newsletters and other communications about this. And if any of you are interested in presenting in our future webinars, please reach out to me and I can definitely accommodate your requests as well. All right, so that's it from today. And thanks once again, everyone, for joining. And I hope you have a nice rest of the day.
Thanks, Miwa.
Thank you. Bye.